Thank you. Uh, there won't be Zoom next Tuesday. Don't expect to see it. That'll be for next Thursday. The test is entirely on Canvas. It'll be, I will tell you where it is again, but I'll tell you now it'll be in the module for that week. Okay. And usually I put it at the bottom and it'll say practical exam one, and you won't be able to open it, or if you try, you'll just get a message, of course, that says the exam is available at 2 o'clock. Okay. And the exam is 100 questions. You do have some extra credit available to you. The extra credit that you have available is 1 point for each of these Zoom conferences that you've attended. Okay. So you got 1 point today. It works out to be about just the same, except that you don't have to answer special questions in order to get it. And I accomplished what I want, and that's have you listen at least once. Hopefully, I'm not always boring, but I feel you gain a lot in listening to it. I know some of you are looking at it on the recordings. Okay. And as we get closer to the exam next week, I know what'll happen. More people will review the recordings. Okay? You don't need to review the recordings because all of the information that'll be on the exam next week is on Canvas. Okay? All the information for our lecture test when we get to it, not next week, but when we get to a lecture test, that'll be all on Canvas. But for the practicum, it's 100 images of bones, and each one will have a single label or question. If it's a question, it may say, name this bone. That's actually pretty easy, except for a couple of bones. If it's a structure, I always use the mastoid process as an example, the styloid process. I could use the perpendicular, harder one, perpendicular plate of the ethmoid bone. Then there'll be a pointer right exactly to that structure. If it's an arrow that I use, then the very tip point of the arrow, where it lands, that's the structure that's being asked. They're not all arrows. Sometimes they're just lines, and that's all they need to be. Okay. The material for the practicum is in module 1, 2, and 3. Labs. Every day that we've had, there'll be 5 labs. 2 in module 1, 2 and 2, and this week we'll only have 1. That's today, and Thursday we'll have review. Okay, so plenty of time to study, ask questions, but no new material. And it will come from those items in the modules that are labeled lab. You'll notice or recall that most of the things are labeled either lab or lecture. Okay. For the practicum coming up, our first one anyway, things may change slightly later on, but for this first one, it's going to be those module items that say lab. Okay, so they are the skull. There are 2 of those, 2 links to the skull. They're the vertebral column, upper appendicular skeleton. And now today we have to do the lower appendicular skeleton. And I have to talk to you a little bit about x-rays, because some x-rays are going to be used on the test. And don't freak out. Don't freak out at all. The x-rays are clear. They look just like the bones, except that you can see through them. In reality, on the job, if you're in the health field, you're not going to be looking at bones. You're going to be looking at x-rays more often than not. If you see a bone, it's a compound fracture, and I hope you don't see too many of those. So there are those 5 demonstrations called lab that show the bones and have labels on them, and there are recordings to go to each one. Prior to each one of those, there's a list of locations in the book. It says you can find the parietal bone, for instance. I'm just making this up. The parietal bone on figure 1-1. Okay, and it was directing you to that so that you can find it easily. But that list, all of those lists, are the sum total of the bones that you have to learn. Okay? So although they're listed out and identified with a figure next to them, that's your list of bones. That's your master study guide. Okay? There will be no other types of questions next Tuesday.
I have a question.
Thank you.
Will there be a discussion section after the test to discuss?
Let me think it through, okay? There'll either be a discussion section before the test, possibly, or after. It won't be both. And my experience so far, but not with you as a group, is that afterwards, the only thing people want to talk about is try to get credit for a particular question, okay? And I do that so quick. I mean, if there is an error on the test, I get it fixed for everybody. And nobody really has to ask that. They email me, and so I don't know if I want to use the discussion time for that. Okay. One of the discussion questions in Canvas in our class that I have usually used is, did you think that your grade was fair, proper, and accurate when all was said and done? And unanimously, everybody said yes. You know? So I will be fair.
Thank you.
I told you once before about this desire I have for justice in my life, and I've had it quite some time. And to be fair is very important to me. I told you about the guys that threw rocks at my car, and I went back and I deflated their situation. That was when I was an adult, but even as a child, I would have to have fairness. I grew up in Los Angeles, and I had a sister 1 year younger than me, and it was kind of boring, nothing much to do. And you had to find the other kids and make up games. We didn't have real toys or anything like that. Down the back alley of our house, where the houses on either side, you go down the alley, and my God, the grass was really parched and about 1 to 2 feet high. It really looked like a fire trap. And that was okay. That's just the way it was in those days. We went down, my sister and I, to Kathy's house. Kathy, she didn't live too far away, a few houses down. And we asked Kathy, does she know a game we can play? We want to do something. And everybody batted around ideas, and we had no good ideas. Finally, Kathy says to us, I know, I'm going to get into the uh, cupboard over there. The cupboard was attached to a detached garage, an old-fashioned garage. I'm talking about the 50s here. And you can lock me in and count to 30 and then let me out. And then I'll put you two in and close the door for 30 and let you get out. Now, obviously, that would not be $1 million seller for Mattel or any other game these days. That's dumb. But to us, being bored, sounded okay. So we put Kathy in, held it shut. There was a lock. We didn't use it, and let her out after 30 seconds. Asked her if she was scared or whatever. No, she wasn't scared. She put the two of us into this cabinet, and at the end of 30 seconds, guess what? Kathy didn't let us out. Kathy had applied the padlock. Kathy had us trapped in this small cupboard, and we were panicked. We were screaming our heads off, and that's not right. We kicked and kicked and kicked, and finally we broke out of the cabinet. We broke out of the cabinet, and I looked up to the second story of their house, Kathy's house, and there she is in her bedroom window, and she's just laughing her head off at us. She's laughing her head off. Now, to a 5-year-old that believes in justice already, that was not acceptable. That can't be. Must compensate for that. I didn't use words like that when I was 5. We went home, and I sat and thought about it for a moment. And I got a piece of metal, a rod, and I poured oil on it. I got a book of matches. I was allowed to have matches at 5 because I was in charge of the incinerator. Our houses in Los Angeles contributed to smog by incinerating our own trash in the backyard. And I was in charge of that. So now I go down to Kathy's house, through the back, and into their yard, which was nothing but tall grass, horrible, dry grass. No one's around. No one's around. I started the fire in the incinerator that they had. And when it got good and hot, I put the metal rod in, pulled it out, and I had a torch. And I took that torch, 5-year-old kid, and ran through the backyard, ran through the tall, dry grass, and lit their house on fire. Didn't get to the house, fortunately, but this was payback. And I went home, sat on the porch. The fire trucks came. My mother said, "Go out and see what the trucks are here for." And I said, "No, I don't want to." He said, "What do you mean you don't want to? That's a fire truck or something out there. Go and see." I said, "I don't want to." I obviously knew already and didn't want to get caught. So I did finally take a peek. The garage burned down. The cabinet burned up. The grass, the backyard, all completely gone. Fortunately, their house was okay. We got word that Kathy got sick, and very sick. We went over to her house. She wasn't in a hospital. There was a doctor there that came to the house, and we commiserated, if that's the word, and asked her how she felt and everything. It turns out Kathy almost died. She had been poisoned, and my sister had poisoned her. My sister believed in justice too, but a little bit too extreme. My sister took an oleander branch, deadly poison, put it inside of a garden hose, and rubbed it and rubbed it and scraped it because Kathy always drank out of that garden hose. My sister's only 4 years old and she's doing this. Anyway, Kathy survived, and we felt that we had our justice. I'll be fair on the practicum. If there's a question that needs to be fixed, do not get all excited. It will get fixed. If there's a question that you think needs to be fixed, and I know it doesn't, know that of course it won't. Any other questions about the practice?
I have a question.
Yeah.
Do we get to know what we got wrong and what we got right after we submit it?
Yep.
That's it. Okay. Thank you.
Yeah. As soon as you submit it, you'll see your test, and it'll have marked for you what you missed and what I had put down was the correct answer. Okay. Yes. Do not look or believe the grade Canvas will tell you you have. It probably will not be enough because it doesn't incorporate the extra credit. Okay. So I won't even entertain a question if anybody says, my Canvas grade, it says that's enough. If you say my Canvas grade, that's the end of the discussion. We don't need to talk about the Canvas grade. Okay. Now, you're welcome to use some time here to talk about that exam or anything else.
I have a question.
Sure.
We'll have the whole time period to take the test.
No, you have, I'm giving you too much time, believe me, but I'm going to give you an hour and 20 minutes. That's standard for anatomy. Okay, that's the standard length of the practical examination. Always an hour and 20 minutes. I really feel that the bad part about giving you an hour and 20 minutes is you don't need it first off, and then you tend to second guess yourself. Okay. If you second guess yourself, you usually guess yourself wrong. Okay, but that's your prerogative. You have an hour and 20 minutes. Okay.
Charlie, I have a question.
Yeah.
So we only need to know the terms on the list, right? The lab list.
I'm trying to think if there's even an exception. On this first test, I don't believe there's an exception at all. You're right.
Okay, because earlier you mentioned, I think it was styloid process, but that term is not on our list. So I was just checking.
Okay, well, what I'm going to do is, of course, I do it anyway. I'll retake the test myself right away. And if there's anything that I need to inform you of, I've got Thursday to do that. Okay.
Okay. Okay, thank you.
And if you're not here Thursday, there'll be a recording to tell you. Okay. There's not going to be any surprises. Okay, nothing else? Okay, well, we have another chance around the break time and such. I did some checking and calling, and because I didn't like the stuttering that we get occasionally when we go to share a screen. Once we even got stopped, they say it's common. It happens to everybody. I said it shouldn't happen to anybody, but we may get the stuttering still. Okay, so let's find out. Okay, you're not seeing anything in particular, are you? Are you seeing a time, 2:30, 2:24?
No, it just shows.
No,
Okay, no problem. Okay, I'm just going in the way I know works. Okay, now you're seeing the modules. Who came
Yeah.
up with that term? Module. Module. I don't like it. I'd rather just call it week, you know. Okay, here we are for week 3. It's short looking, but of course, you know, we're going to add recordings to it and other material. Okay, and I will be talking to you about the X-rays a little bit today, but mostly on Thursday. I do not consider that new material, but you might, but I don't. Okay. The discussion, it's this time I am going to ask you again to make another question. I don't usually do this, but to get started, I would want a question. This time, though, I want an image. I don't want just a text question. Okay. I did read your questions last week, and I inserted my comments once in a while. I think one of them was wrong, so I had to insert the comment so that everybody knows what the correct answer is. But so for week 3, what I'm looking for here is like one of the images that I show you, and then label it. Okay, now sometimes you can label it with words. What is the projection behind the ear, for instance, mastoid process? But you'd have a picture of the skull with the mastoid process. How are you going to get this image? You're going to just look at the internet. Put in there, for instance, if you want skull, put bones and skull, and then click search, then click images and pick one. Cut and paste, cut and paste, or save it to your desktop till you're ready to do that. Okay, and sometimes you'll very likely find images that already have arrows, and you can use those to make your question. Okay, I want you to try to make a question like what you see when I go through the lab with you. If you don't remember that, I'd be surprised, but you'll see it in a moment when I do the last section. The assignment for this week is chapter 2. Chapter 2 is the cell, so we can get it over with. The cell chapter can be daunting. It has so much stuff, and I'm going to go over it with you right now. Okay.
Charlie?
Yep.
How can I submit the assignments? I send it to you by email?
Yes.
Okay,
Okay.
thank you. Okay.
Okay. Outlines are sent by email.
Okay.
Do not use inbox. It used to work nicely, but too many people get too confused, and I don't need that. You don't need that. We all know how to use an attachment, I think. Okay, so please send anything to me that way. Do not send me an email message like, oh, I'm going to be late to class today. First off, I don't need to know that. But if you wanted to send that, send it by email. Don't send things through the inbox. Okay. It's it's hell to retrieve them. I used to like it, but I really don't now. Now the discussion that we just passed, and I asked you to do an image and to make a question, that you paste right onto the page in Canvas. And in doing that, then you reply and it posts. That's all you do. It's already in there. You don't send it to me. Okay. The only things that you need to send to me for the entire course are the chapter outlines. That doesn't mean you don't need to contact me because you're lost on this or that or want some clarification. Absolutely, do that all the time. Okay, so for chapter 2, and if you don't have the book in front of you, again, you can come back to this later when you do have the book, come back to the recording, or you can just make notes. My chapter 2 starts on page 23. And it has a picture of a cell. That cell is like no other cell in the world. No cell looks like that. You'll never find an exact copy of anything that they put into the book. Okay. They're being idealistic. The only things that you need to know on that diagram are those things that we put on the diagram in the lecture last Thursday. Any new terms you don't need to know. I'm looking for new terms. Centriole, we haven't touched that yet. We will. Okay. Almost all the other terms we've hit. Peroxisome down at the bottom right. No, you know. If this were my book, I'd put parentheses around it. Okay. Okay, then text-wise, overview of the cell. Yes. That entire thing. The diagram, figure 2.2, no, you know. The plasma membrane, yes, pick up the reading there. And now it's interrupted by table 2.1, which lists the parts of the cell. Yes, for table 2.1. Pay attention to those things that we talked about. But again, peroxisome is here, and we haven't talked about it yet. Microfilament is here. We haven't talked about it yet. We will be talking about some of them. You can add them in later, or you can leave them out. Okay. So that you, in case you want to get started. The next page, structure, yes. Function, yes. This is a bit more than normal. Membrane transport, yes. The cytoplasm and cytosol, yes. So you see I'm not skipping much here. The clinical application, of course, no. Check your understanding, no. I'm back to cytoplasm, cytosol, cytoplasmic organelles, yes. Put a check by ribosome, endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, lysosome, mitochondria. Do not bother putting a check near peroxisome. You can put that in parentheses or just say no. Okay. Cytoskeleton.
Sorry,
No.
Charlie. Peroxisomes, yes or no?
What?
Peroxisome?
No.
No? Okay.
Cytoskeleton, no. Centrosome and centriole. centriole, yes. Centrosome, no. So I'm going to split this paragraph here. What it means is I would already test you on centriole. We're going to talk about the centriole enough on Thursday. Cytoplasmic inclusions, no. The nucleus, yes. Its envelope, yes. The nucleolus, yes. Chromatin and chromosomes, yes. But now I've got a diagram pair, 214 and 215. Okay, we are not in genetics class. Okay, I enjoyed it when I took it, but most of the people didn't. It's easy to forget, and anyway, but this is not part of gross anatomy at all. So the figures 214 and 215, no. Okay. Then the life cycle. I'm going to go over that with you on Thursday, and what I present is what you'll need there. You do not need to outline that part of the chapter. So the life cycle, find it, and write stop above it. And that's where you'll stop. And that shortens the chapter somewhat and gets it down to the material that we really need.
So Shirley, the last thing is that the cell life cycle.
Not to outline, no. That put the word stop above, right above the life
Okay,
cycle.
okay, thank you.
I have a question. I missed it. Did we need Golgi apparatus and lysosomes?
Yes,
Okay.
I think I'm pretty sure I said lysosome. I don't remember saying Golgi apparatus. You know why
Okay,
you know why you need it? It's in my notes. Okay, so when you get to studying for the lecture exam, it's a little bit different. You need to know those notes. You need to know them backwards and forwards. So things like, for instance, here on the screen, where it says intro lecture, joints lecture, ossification lecture, levels of organization and review of cell structure. You need to know these. You need to know these notes so well that you can recite them backwards and forwards. There's not that many. There's not that many. It's not like reading a huge book. Because that's where 95% of the questions come from. 5% of the questions on the lecture test may come out of the chapter, and the important parts that I would have expected you to outline, but I left it for you to include in your outline. They're important things we may have talked about, but there's not many. And I never get any flack for those questions. Okay. What I see on the first test is a lot of people scoring very high for the first lecture test I'm talking about now, but many people scoring in the 50s. That's no good. And it's strictly because they did not memorize the notes. You've got to memorize the notes. You can't do it the night before. And a lot of people I know have the tendency, and I would too. We've got two things going on at the same time. We're doing lecture and we're doing lab. They don't always coincide. We've got 4 basic types of labs and 20 odd types of chapters. You must keep up with the lecture, even though it's the lab that's coming first. Does that make sense to you? Okay. Read it, try to absorb it, and just keep it fresh. If you wait till the night before, it's not going to work. I do get plenty of people that score 98, 99, 100. That's probably about half the class. And the other half of the class is just going to slip away. And I don't want to see that at all. You've got to remember the lecture notes. Okay. If you wait, I'm sorry to harp on this, if you wait till the night before to look at them, you won't be able to ask me enough questions. And I won't be able to answer enough questions. I'll be busy with others. You will not do well at all. Okay. There's no reason not to do well on the lecture test. The questions are word for word out of the lecture material. It's all there. You do not need to go back and reread the book for the lecture test. You should not. Instead, read your outlines and then definitely read what I've posted on Canvas. If you don't make decent outlines for yourself, they'll be valueless and you lose a really important tool for studying. Okay. Okay, today we're at 9/7 and our lecture is cellular processes. A process is anything that goes on as a sequence. This causes that, that causes something else to happen. Okay. Many things are considered cellular processes, like cell division, DNA replication, but only 6 of these processes involve only the movement of water and salts. And these are the ones that we're going to be talking about. The movement of water and salts within the cell, within and without the cell, between cells. There are 2 types of processes, active and passive processes. Passive processes, passive movement of water, passive movement of solids, these do not require work. These do not require work by the cell or the organism in which they occur. Well, for any process in the universe to occur, there's going to have to be some energy expended somewhere. But we have what we call Brownian motion, okay, and kinetic energy that we stored from the past. Passive processes are not going to use any special amount of energy to proceed. Well, when we talk about them, we understand it. And the first of these 4 passive processes is so ingrained in all biology students, we shouldn't have much trouble. It's diffusion. Okay, everybody knows real life examples of diffusion. It's a movement of an area of higher concentration to lower concentration. And that movement from higher to lower concentration continues until an equilibrium is reached. Lots of non-cellular examples here. Open a bottle of perfume. The perfume inside is very, very strong and concentrated. There's none outside until you open that bottle. And then you get to liberate some of the vapors. and the perfume keeps evaporating out of the bottle, even though it's going to be slowly, until it's all gone, or until it's equal in the bottle and outside of the bottle. But if you're in a closed, small room, the perfume might evaporate. If you've got a large mouth bottle, the perfume might evaporate rapidly, more rapidly. But it will stop when the concentration of the perfume is equal in the bottle and outside the bottle. There will still be perfume in the bottle. We can think of diffusion as the movement of salts from higher to lower concentrations as well. Salts, any sort of a solvent or solid. The universal solvent is water. So let's use water as an example. Think of a fish tank, rectangular, big old-fashioned fish tank. That's filled with water. That's our solvent. And now take a dropper full of vegetable coloring. I like blue, so I'm going to put a drop or two in the top corner of the fish tank. It's going to do what? Is it going to move out? Is it going to disperse?
It will disperse.
Right. Dispersion is another name really for diffusion. Okay. How long will it continue to disperse that blue color?
Until it's equally distributed.
Until it's equal everywhere, until equilibrium has been reached. How long do you think that might take? Somebody else, how long do you think that might take? Fish tank, about 1 or 2 feet across. As far as I'm concerned, it's going to take forever. It's going to take as long as it takes me to get an answer out of you guys. Okay. And here's an example here of a dye being pushed into water, spreading out until it reaches equilibrium. Okay. That is simple diffusion. Diffusion happens everywhere, inside the cell, outside the cell, but it doesn't happen through the cell. Diffusion does not leave the cell if it's inside happening or outside happening. It does not cross the plasma lemma or the membrane. I think if my biology teacher had made that clear, I wouldn't have had so much trouble with the second one. But now it's a snap. This is osmosis. Osmosis is one of those terms you heard about, studied for, did well on the test possibly, now you've forgotten it. We all get to where we are on the basis of what we once knew and may now have forgotten. Fortunately, we haven't forgotten everything. We haven't forgotten the stuff that we need to know on a daily basis. Osmosis. It's a special type of diffusion. It's diffusion through a semi-permeable membrane. Something's being allowed to cross the membrane. Something's not in one or both directions. Come into a room. I always think of a yellow, old-fashioned room. Just a square, boxy room. Got one door in, and you come in, and you've got perfume bottle and a jar of flies. There's a window, a simple old-fashioned window, 2 shutters and a screen, and the screen works. Close it all up, close the door, seal the bottom of the door, just you and those 2 bottles in there. Okay, if you open the perfume, it's going to diffuse into the room. If you open the jar of flies, what will they do eventually? May take a little longer. Will they diffuse into the room? I don't know if we'd use the word diffusion, but they certainly would disperse into the room. Okay, so pretty soon I've got flies, perfume in the room, and I'm wondering what I'm doing there. So I go over to the window. The window's been closed, 2 glass shutters, and I open the window, and there's the screen. The screen's in good working order. Can the perfume now continue out? Yes, the perfume will go out. Can the flies get out? Presumably, no. Flies are stuck in there. The screen is a semi-permeable membrane. Some things get out or through it, and some things cannot. The plasma lemma is a semi-permeable membrane. In the cell membrane, the primary example is the plasma lemma of a semi-permeable membrane. Sometimes things can enter to the cell, like amino acids, and once inside the cell, form proteins. And those proteins can be incorporated into many things, including chromosomes. Well, it's like building a ship in a bottle. You put something small in there, build it up, and you can't get it back out again. The chromosome's not coming out. The DNA should not be coming out either. So this is a semi-permeable membrane, the plasma lemma. These are blood cells, red blood cells, and they're all in some sort of plasma. Okay, that's the fluid portion around the blood. We'll start with the one in the center. The one in the center, the plasma membrane around the red blood cell, is semi-permeable. But in our example, for salt and water, it's going to be permeable to both. So we have a concentrated solution in the plasma out here in the black of vital salts and water. If the concentration inside of the red blood cells is the same, then there's no net flow. We can have things move in and out, but the move out is equally as they move in. And everything stays looking the same. This is called an isotonic situation. Iso meaning the same, tonic meaning strength. The same strength on both sides of the red blood cell membrane. So water comes in and water comes out. When I was taught this, and the word tonic means strength, and it always referred to the salt or the solid. But now they have changed it within the last 10 years, and the isotonicity refers to the concentration of water. The movement of water in this case, is equal to the movement of water out. And the red blood cell stays properly inflated. It's possible also that you have a hypertonic solution. Hyper, high, tonic, strength, more solute out here. In other words, add salt to the mixture. And literally think of taking a salt shaker and shaking it. And what will happen is the water concentration will be greater inside than outside. The solute concentration will be higher outside than inside. The solute concentration is hypertonic. That's where that word hypertonic comes from 10 years ago. But now it means when we have a situation where the water is greater inside, a higher concentration of water inside. And what will happen is the water moves out quickly from the red blood cells out into the plasma to dilute the plasma back down. But in doing so, it crenates. The word crenation means to make into a smaller cell. It shrinks. These cells do not function very well. These cells are caused because there's a high concentration of solute, and that doesn't necessarily mean salt, but any mineral in the plasma. How the hell do you get such high mineral content in the plasma of your blood? You're gonna have trouble if you did. Well, in some places they inject it. They inject it into you and cause the collapse of the vessel itself to get rid of the varicose veins. By injecting salt water, into a varicose vein, it literally will disappear from the surface. You need to know the word crenation. I'll look for it in just a second. Isotonic, no change. Hypotonic. Hypotonic solution. Hypo means less strength, less solute out here. But that then means more. If there's less solute out here, there's going to be more water. Less solute means more water. And the water is going to rush into the cell. You could wait, if nature would, for the solute to move and redistribute itself and reach equilibrium, but that takes forever. Water moves immediately, and the water will move in a hypotonic situation into the cell. And in most cases, with the red blood cell, literally blow it up and burst it. That's called hemolysis. Hemolysis. These are all examples of osmosis, movement through a semi-permeable membrane. Hypertonic solution causes crenation. Hypotonic solution causes hemolysis. Okay. Here are is the text for each of those. Isotonic, no net movement of solvent. Hypotonic, higher concentration of solids inside the cell than outside the cell. When it occurs, more solvent will enter the cell than leave it, and that can result in hemolysis. Hypertonic, more solid outside the cell than inside it. More solvent will exit the cell will shrink, and that's crenation. Facilitated diffusion is the third of the passive processes. Facilitate something, you help it along. So we're going to help diffusion along. Diffusion proceeds at a particular rate. And if this is a cell membrane here, and something is diffusing in, it might diffuse kind of slowly across. But the cell membrane has what we call protein channels and carrier proteins. And each of these can facilitate the movement of a solid quickly inside. Now it's not using any energy to do this. It's a passive process. Just like a whole bunch of people coming to a gate or a wall, and I'm not trying to be current here at all, but finding a large opening in the wall, go through fast, go through slower going over the wall. Facilitated diffusion speeds up the diffusion process faster than would be mathematically calculated. Take that fish tank that we had a moment ago, and the blue dye is now making its way slowly across the empty fish tank. Well, let's do something. Let's put a goldfish in. Dye won't hurt the fish. Okay, the fish is going to swim, and in swimming, it's going to stir the dye in the fish bowl. That's facilitated diffusion. When you get out of an airplane, you are stuffed in there usually. Now, I haven't been on an airplane now for a year myself. Before that, I've been on 400 flights and love them all, but not today. But when you get off of an airplane, you get out of that cabin, and what a relief usually, a cattle car and lots of people. You get down the ramp or go through the tunnel, and then you get out into the terminal and you spread out. Everybody spreads out. Okay, well, that would be expected. And then you find the speed ramp, and you get on the speed ramp, and there you go. That's facilitated diffusion. Okay. Unfortunately, that particular process comes to an end when the speed ramp reaches baggage claim or passport control. Dialysis. Boy, I don't have a lot here to say about dialysis until we get to the kidney. Dialysis is the passive separation of molecules by size. If somebody's undergoing dialysis, they remove his blood or her blood, and fraction the contents by their size into different pouches, and then withhold some of the pouches and return others, trying to withhold the pouch that would have the greatest amount of uric acid or other unwanted waste products. Okay. Those are the four passive processes: diffusion, osmosis, facilitated diffusion, and dialysis. We'll be talking a lot more about dialysis later. Active processes are those that require work, extra work. Why the work? Because they're going to concentrate something. Anytime you concentrate something, whether it's bricks or money, you probably have to do work in order to do it. Okay. We as organisms here are part of the universe, and the universe is essentially running downhill. Okay, in other words, becoming more and more dispersed. Yet we're not. We're not becoming more and more dispersed. We have, for instance, a higher concentration of iron in our bodies than in the area surrounding us. We have accumulated it. Okay. Oh, that wouldn't be true if you're sitting on a pile of nails, but I know none of you are right now. Okay. Calcium in your bones is accumulated against a concentration gradient. In other words, coming from an area of lower to higher concentration. This is opposite what the universe is doing. But we are essentially a speed bump in the universe. We will give it up when the time is right. Okay. So, active transport. We organize our environment. We have a higher concentration of many things in our body that are found in the outside world. Okay. And then there's two examples that I just gave you. To get higher concentrations of solids into our body and keep them there, we must expend energy to pull these things into our cells. They just can't go against the concentration gradient. They can't come into the cell if there's more of something already in the cell. We are taking things from an area of lower concentration to higher concentration, and we are not in equilibrium with our environment. We again have carrier molecules. Okay. But in this case, something different's happening. A smaller amount of a valuable item, nutrient, protein, amino acid, smaller amount is still being pulled in and added to a larger amount. So we're going from an area of lower to higher concentration. In the other one that I showed you here, this was facilitated diffusion. We were going from higher to lower concentration. That doesn't take energy. This takes energy and utilizes ATP. Where are most of the, if you think of a prison yard, and all the prisoners are out in the yard, you've seen pictures of that, okay? They're highly concentrated right there. They're more concentrated than they are outside of the prison. But where are most of the people who should be in prison, do you think? Inside a prison now or outside? Well,
Inside.
think they're inside? I think most people think they're outside, actually. They're still here with us. Okay.
Charlie,
Yep.
the bottom, all the blue dots on the bottom, that's showing the outside, right? The top is the inside.
I'm sorry. I got distracted by the sound, so I muted it.
Oh,
I
that's okay.
I went hunting for it and may hopefully found it. Okay, so now would you ask the question again?
I was just saying, is the model with the blue dots on the bottom representing the outside, and then the lower concentration is on the inside?
No. This is the outside at the top.
Got
Good
it.
question.
Thank you.
Yeah. Good question. Bad diagram.
So is that for all the models then? Same thing? The outside is
Yes.
top?
Yes.
Okay. Thank you.
Okay. This is showing a smaller concentration on the outside than inside. Well, when a prisoner is brought to a prison, they're not brought in large amounts normally, and they're going to be put in here for the general population, and there's going to be a lot more crowding. That takes energy. There's no prisoner I know that's going to want to walk in. They're going to be taken in. And then probably their first line of first duty is to try to find a way to escape. And they'll find a way to escape. And as they try to escape, they'll be pulled right back in by the guard again. So it takes energy to keep the solutes, or in this case, the prisoners, still inside. Now, some prisoners or solutes will eventually get away and they're gone. But other ones are coming and being brought in so that they increase in amounts. Cytosis, we already know this one. We've mentioned it already. Cytosis comes in two phases. Phagocytosis, which was cell eating, and that happens when a food vacuole is formed and a food particle is brought in from the outside of the cell. Remember the golf ball with all the dimples pulling things in. If it pulls in a solid piece of material, it's phagocytosis. That takes energy. If it misses that but still gets valuable liquid material, it's called pinocytosis and forms a pinocytic vesicle. Both of these are active processes. Okay. Now, I'm going to give you a break, but any questions before?
I have a question.
Sure.
So ATP is the energy causing active transport.
ATP doesn't cause active transport. ATP allows active transport.
Yeah. Okay.
It's sort of like gasoline doesn't make the car run. You do, but you can't do it without the gasoline. It's the fuel.
Thank you.
Thank you. Okay, it's 15 after that last part. Take a break till 3:25. Bye-bye. Okay,
Seriously.
okay, anybody come up with any sort of a question?
The lecture seemed pretty straightforward.
That's the way it's supposed to be. I'm gonna butt in where I'm not wanted here because I'm concerned. I notice you are looking into starting study group. I think that can be a very good idea, but it can also go terribly wrong. So I just want to warn you, don't let it go wrong. Okay, what can go wrong with the study group? Okay, well, I'm going to say it because I don't know any of your abilities yet. I haven't seen your test scores and everything. You are going to be supporting the person who probably is doing the least amount of work. Okay, there's always somebody in the group who's doing much less than others. And I don't know how do you going to stop that from happening without being blunt about it, or just people end up quitting because they don't want to see that. They also take up a lot of time. But for some people, study group is good, and there's nothing social that we can do anymore. I mean, so that might that could be very very helpful to some people. Okay. Just don't let anybody drag your study group down. I mean it. Okay. And if you are going to join a study group, be and not participate, but only take away, then maybe you shouldn't join the study group at all. And the opposite is true for the people who are going to participate. Then I think you have a good chance of helping yourself out. Okay? Now, do I think you can work without a study group? Sure I do. But maybe that's not as much fun. Okay. And maybe if you're well studied, you can help somebody else along, and that's good. But what I said is still goes in my mind anyway. Don't let anybody drag your study group down. Okay. You didn't want to say anything, so I did, and I don't want to debate or anything. Do you have any other questions though? Okay, and back we go.
Will we go over cell division on Thursday then?
Yes, it's
Okay.
already scheduled for Thursday.
Oh, thank you.
I also am going to talk, as I said, on Thursday about x-rays. But if you go to my website, go back to module 1, Charlie's website, get it on, and you can find x-rays right over here. Okay, under unit 1, that's where we are. We're finishing it off. Okay, and you click on that, and there are x-rays, and they will have the names of the structures. You haven't seen this one because this is part of today's lecture. On the other hand, let's get one that you have. Okay, here's a shoulder. And you look at that, and you can get a labeled version right here. But obviously, this is the head of the humerus. This is the glenoid cavity or fossa. Okay. Coming from the front, this is the clavicle. Coming from the back, we can't make it out quite as well, but the acromions back there would never ask it from this side. This little guy. Well, when you look at your book and we look at your notes, you'll realize that's that broken little finger I talked about. That's the coracoid process. The one thing about an x-ray that you need to understand is that you see right through it. There is no front or back, really. There is no right or left. Okay, here we go with this guy. This is the ulna. You can't see it here, but it's going to go to the side of the little finger. And the radius, here's the radial neck tuberosity. That's not, that's fine for me, but that's not good enough to ask you. The round structure here is the capitulum, and the mouth on the ulna is the trochlear notch. Let's try the hand. Okay. We have distal, middle, and proximal phalanges, or just the distal and proximal of the thumb, metacarpal 1 through 5, trapezium, trapezoid, capitate, hamate, and here's the pisiform. Here's the styloid process of the ulna. Here's the navicular or scaphoid. This is the lunate. And here we see we've got a buildup of bones here. We've got the triangular in here. We've got the pisiform, and we've got a little bit of the capitate. When I look at this more closely, like I was second guessing a question, this is the triangular or triquetral.
Hey, Ann, there's some spots on here that weren't for sign off. So this one didn't have a thing, so he didn't sign.
Well, he hasn't been there yet.
Yeah, he has. See?
Okay. Trapezium, scaphoid or navicular, lunate. Here was the triquetral, triangular, triquetrum. And see underneath it here in a circle, that's the pisiform. Don't worry, I'm going to be straightforward. I'm not going to get any that take a radiologist to figure out. But what I'm saying is these are no different really than looking at the actual bones, but this is what you'll see in real life. I'm showing them to you now, and I'll go again on Thursday in case anybody wants to take a look at them, make a head start, or if you find one that's just impossible for you to figure out, well, you can ask me about it. Okay. Okay. These are the bones and figure locations for today. And so somebody was asking me earlier, are these the structures that you have to know and the bones that you have to know? And that's exactly right. Okay. It always seems like so many, but you're going to have 4 or 5 days more to study after Thursday. And Thursday, I'm not giving you anything new as far as the bones are concerned. Okay, so here's the figure pages. Figure pages for today look like 8-7 is tops, 8-9, 8-10. Up, I often hold off on the knee until Thursday. Okay, then the next one for lab almost looks the same length, but if you read it closely, it's not. Okay, upper appendicular. No, I don't want that. I want the lower appendicular skeleton. I'm sorry. Okay. Okay, you might just call this the hip or something, but you can't get away with that on the exam at all. If you feel the top of your, well, call it hip until we can get another term. If you can feel the top of the hip behind your back on either side, okay, that's the first of 3 bones in there. There's 3 bones called the ilium, the ischium, and the pubis bone. Sometimes it's called the pubic bone. Doesn't matter. The ilium here, okay, is the first of the 3 parts of this whole bone, which is referred to as the coxal bone by the book and anybody else, COXAL. However, that will not be a an answer on our exam at all. Okay, we start with the ilium, and if I put a pencil or my finger at the top, that's the iliac crest. Now, we're going to have to figure out where's the front and where's the back of this bone. Where's the anterior? Where's the posterior? Well, it's kind of easy when it's labeled like this, but come this way to this spine. This is the anterior superior iliac spine. And if you were able to drop 1 inch or so below that, It's the anterior inferior iliac spine. Now, at the beach, you have seen somebody in a swimming suit. Man or woman makes no difference. They've got two knobs on either side. Two knobs on either side. They're not unattractive or anything like that. They're just there. Okay. People say you hitch your belt to that, but not your swimming suit. Well, these are the anterior, superior, and inferior iliac spines. Now, go to the other direction and come to the end, and it's the posterior superior iliac spine. If you wanted to find the posterior superior iliac spine, you're going to have to go to your back. And if you have a sore back, it'll be right there. If you've ever had a sore back, because this articulates with the sacrum to make the sacroiliac joint. This is the posterior superior iliac spine. Drop down 1.5 inches. The posterior superior iliac spine. Okay. Where's the back? Where's the front? Okay. This word ilium would be printed right here on the posterior of this bone. And the front side, not showing, over on the other side if we turn it around, that would be the anterior. Here's what you're looking at real quick. This is the anterior that we didn't get to see. We're seeing from the back side. This is the anterior superior iliac spine. Anterior inferior iliac spine. Back here is the posterior superior iliac spine and the posterior inferior iliac spine. Okay, so here's the two knobs here and the two knobs over on this side. Put your finger on the posterior posterior superior iliac spine, drop down to the inferior, and there's a big gouge. That's the greater sciatic notch. If you put your finger in the greater sciatic notch through which the sciatic nerve does run and come around, you come to a point. This is the ischial spine, meaning you're in part of the ischial bone now. It really goes from here straight across. Here's the ischial spine. Right underneath the ischial spine is a small groove. This is the lesser sciatic notch. The bone is labeled in black, ischium, but there's a solid and fairly large bump here called the ischial tuberosity. This is what you're sitting on right now. Two of them. If you've ever sat on bleachers, you felt it. If you've ever got saddle sores, it's from riding and bumping up and down on these ischial tuberosities. And if you've sat on bleachers, Same thing. This is the ischial tuberosity. Come around underneath this hole, and this lowermost part and anteriormost part is the pubic bone. The hole is the obturator for Raymond. Obturator, you probably never heard the word before. There's nerves called the obturator, muscle called the obturator, and they all pass through here. And then you have above that a concave depression, just about perfect for a golf ball, but obviously meant for the head of the femur. This is the acetabulum. There's some pretty outstanding features here, and they've got some pretty outstanding names, unfortunately. On the anterior side, this part of the ilium is the iliac fossa. The pelvic brim is labeled over here. Goes all the way around. And then the male is smaller, since the male does not have to give birth. This is the pelvic brim. The best way to tell a male versus a female pelvis is to look at the angle here where the two pubic bones come together at the pubic symphysis. In the female, it's shown here as 100 degrees or more. In the male, less than 90. I'm going to tell you, I've never seen it 90, and this picture does not show 90. I'd say closer to 60. This is definitely not a right angle, though. This is definitely bigger than a right angle. Here's the ischial spines that show, just mentioned. In the female, the ischial spine size is important because it and this structure, the sacral promontory that we talked about a week ago, they penetrate into the pelvis and can make it difficult for an infant or newborn's head to get through. Now we're going to insert the head of the femur into one of the acetabula. This one. Okay, this happens to be a right, no, I'm sorry, it's a left femur. How do I know? Well, first let's just take apart what we are certain of. That's the head. That's going to be the neck. Same on the other side, head and neck. There's a indentation in the head right here, and it barely shows right here. It's not deep, You couldn't even put a pencil eraser into it. That's the fovea capitis. Fovea capitis means hole in the head. Here's the neck. This is anatomical view here on the left. How do I know? I come down to the distal end and it's smooth where the kneecap is going to set. Come up, back to it. Then this is the medial side, obviously, because the head of the femur fits in. On the medial side is the lesser trochanter. And on the lateral side is the greater trochanter. Lesser and greater definitely do mean size reference. Turn it around. You'll see on the back side the greater and lesser trochanter. And like a moon or a banana, joining them is the intertrochanteric crest. Muscles of the leg attached to the trochanters. Muscles in the abdomen coming out to raise the thigh, to flex the thigh, will also attach to the trochanters. On the posterior side of the diaphysis of the femur is this raised portion called the linea aspera. We had deltoid tuberosity in the femur, but we have the linea aspera here. This is a point of attachment for all the hamstring muscles. Let's come down, starting with one on the left, to that smooth surface. That's the patellar surface. You'll notice these two bumps on either side. Turn it backwards, turn it posteriorly. Let's go to the medial side because here's the head and come down to here. This is the medial condyle and the lateral condyle is over here. These two things that almost look like big toenails, these are the medial and lateral condyle also. In between is the intercondylar fossa. Remember, we had the olecranon fossa or olecranon fossa in the humerus. So there's a lot of similarities. They don't look the same, but the way they're formed. On the left, we have a lot of repeat. Let's do it quickly. Head, neck, intertrochanteric line. I won't ask you that one. Okay. Lesser trochanter, lesser trochanter. I won't ask you gluteal tuberosity. How do you know what I'm not going to ask you if you're not writing this down? Because it's not on the sheet. It's not on the sheet ahead of you. In the link that's right ahead of this. If it's not on there, I can't ask you. down at the distal end. Here's the lateral condyle and patellar surface. This is the medial condyle on the medial side, lateral condyle, intercondylar fossa. And now we have the lower leg, the tibia, and the fibula. The fibula is always lateral. The fibula is always lateral. Okay. At the top of the fibula are two condyles, here and here. The lateral medial condyle of the tibia set on the lateral condyle and medial condyle of the tibia, the fibula and the tibia, they match up. If I didn't have the fibula there, and I often don't, here's the medial condyle. How do I know that? Because I'm going to go down and look for this structure that I can barely hold on to with thumb and index finger. That's the medial malleolus. The inside of your ankle. You've hit it before. You've got it sore on a pair of knee shoes. Okay. The inside ankle is the medial malleolus. Fortunately, it's not an oral test because there's so many ways people use to pronounce malleolus. Here's the tibia above it. The anterior border is the front of the tibia. So if you hit your shin right in the middle of the tibia, that bone right in the front, that's the anterior border. Don't worry about the membrane except to know that it's amphiarthrotic. We talked about that. Come down from the top of the tibia, straight down the middle front, and there's a bump. That's the tibial tuberosity. The tibial tuberosity gives rise to some unpleasant expressions, sometimes knobby knees or something like that. But that's just connective tissue that may be attached, more in some people and less in others. This is the tibial tuberosity. It will anchor the patella. At the very top, there's a crown. Looks sort of like saddleback majestic beak. This is the intercondylar eminence. Intercondylar eminence. When it's shown to you in place, here's the head of the fibula, and at the other end, it's called the lateral malleolus. So your ankle on the outside, that's the one that gets a lot more abuse actually, especially with a new pair of shoes riding up against it, for guys anyway. Here's the lateral malleolus. These two form an arch that will hold the foot, much the way that the styloid processes of the radius and the ulna did for the hand. Don't worry about this tibial fibular joint. Okay, that will not be there. That is not on the sheet. Proximal tibiofibular joint is not on our sheet. Here's a portion of the knee, but I'm going to show you the knee as I said on Thursday. But I do want to show you the patella or kneecap. The apex of the patella is actually at the bottom, just like it was in the sacrum. Here's the fibula. Here's the tibia. And the femur. But fortunately for us, the foot, I believe, is a lot easier than the hand. It may be more foreign because most anatomy books, it's the hand they go for. Okay, so for the foot, we have the toes, just like in the hand where we have the fingers. The big toe consists of a distal phalanx and a proximal phalanx, just two. The others have distal, middle, and proximal, all the rest of the way across. The big toe in anatomical position is medial and is number one still. This is metacarpal. Be careful of that, not tarsal. Oh, I'm sorry. I need to be careful of it. The metatarsal 1, metatarsal 2, metatarsal 3, 4, and 5. So none of those are really going to give you any trouble. Now, we've got the tarsals. There are 7 tarsals. There were 8 carpals in the hand, each hand. But in each foot, there's 7. Come back from the big toe, metatarsal 1. And it articulates with just one more bone. And that bone is the cuneiform number 1. First cuneiform 1 is how I pronounce it. The second one, metatarsal 2, articulates with cuneiform 2. The third metatarsal articulates with cuneiform 3. 1, 2, 3. Now, this next bone articulates with both digits 4 and 5, just like in the hand, where we had 4 and 5 articulating with the hamate. Well, this is not the hamate, but it's a bone shared by digits or tarsals, metatarsals 4 and 5. This is the cuboid bone. And the cuboid will come straight back to the heel, the largest of the bones. This is the calcaneus. Sitting up on top of the calcaneus is the talus. It's all of this. The talus is the only bone of the foot that has any articulation with the rest of the body. Move forward and come back to the only one we skipped right here. This is the navicular, sometimes also called scaphoid, just like in the hand. But navicular is going to be better here. Boat shape, upside down, but boat shape. The navicular straddles cuneiform 1, 2, and 3. Okay, now without going over the toes, again, let's just do the carpals. This is cuneiform 1, 2, three, different order. Those are straddled by the navicular, come back and up to the talus. The heel or biggest bone is the calcaneus, and it comes forward to the cuboid, which is also shared by the third cuneiform. Now, if you're looking in a book, and if you're looking in your book, I went to a lot of trouble to find this particular picture. I liked it because it just calls these cuneiforms 1, 2, and 3, or first, second, and third.
Charlie?
Yeah.
I have a question.
Yeah.
We have two calcaneus.
No, one calcaneus, right here.
Okay.
Well, you have two if you count both feet. Yeah.
Ah, okay. Okay, thank you.
Okay. Then they did put in parentheses the words. First cuneiform is the medial cuneiform. It is medial. The second one is intermediate. The third is lateral. But lateral to me would mean it should be out here. And I think that's confusing. So I'm going to use on the test cuneiform 1, 2, and 3. Okay. I think that's a lot clearer to everybody.
Charlie,
Yeah.
really quickly, what did you say about metatarsals 4 and 5? They go along with.
They share a joint with the cuboid bone.
Thank you.
Okay. Okay, I do have the ligaments of the knee. They're right in front of us. It'll save us actually more time than it takes on Thursday. So let's look at the knee. I'm not sure, that's what I'm stalling here. I'm not sure if I gave you a sheet yet for the knee. But in the meantime, you can look it up on Charlie's website under the knee. Okay, and if I did not give you one, you'll have it very, very soon. Okay, here's the patella. Okay, we know that. The patella is held in place. It's held in place by when I'm quiet, I'm usually thinking. I'm ticked off at this diagram. They're calling this patellar tendon when it's actually a patellar ligament. I'll do the knee on Thursday. I'm not going to argue with this diagram of how we remove it. Okay. I just don't want to. I know that would be counter to what is true. First off, and second, of what's in the notes that I've given you. Okay. Okay, anybody with anything? The knee itself is not difficult, but I'm not going to show you a picture I know is incorrect. And I don't know how I must have used it last semester, but
Did we go over, I'm seeing on the lower appendicular skeleton list of figures, I see true pelvis and false pelvis, but I don't remember going over that.
no, you can scratch those.
Okay.
The reason you can scratch them is first off, There's never been a test that asks them. And it takes so long to explain it.
Okay.
And then people forget it immediately. And it's almost, it's more of an idea. And I'd be glad to tell you about it if you want to stay around, or you can ask it on Thursday or whatever. It's just not worth it.
Okay.
Okay. You gotta
Charlie,
try. Yeah.
I have a question about ribs. At the very end of the ribs that attach to vertebrates, what does it called? Is it still shaft or body, or it has another name?
The bone is still shaft or body. And
Okay,
then it converts to costal cartilage.
costal cartilage. Okay, thank you.
Okay. Okay, well, I'll wait around and tell everybody that has a question, had a chance, but you can always, of course, email me. And otherwise, you're free to, you're welcome to go. Canvas takes care of the role for the attendance. So that's helpful to me, at least. Okay.
So Charlie, if we link up our lists, our printed out lists for the figures, and we link up that same, the slides that we just went through, if there are things on the list that are not on the picture, we can assume that they're not going to be on the test, or we just kind of run those ones by you.
First off, if there are things that are on the picture and not on the list, they're not going to be asked. Okay.
Okay.
If
The list is the tried and true.
If you find, on the other hand, that there are things on the picture list, okay, I'd appreciate you emailing it to me real quick.
Okay.
Okay. Now, you don't have to worry about the true and false pelvis, though.
Okay.
That got asked right here.
Right.
It's always best if if you have a question, if you can ask it in front of everybody, I think it gets across better, and it certainly saves time. Okay.
Okay. Thank you.
Okay. So.
Thank you, Charlie, for today. Bye.
Bye.
Thank you.
Thank you, Charlie.
Thank
Bye.
you.
Bye.
Thank you.
Thank you.
Thank you.
Thank
Okay.
you.
Charlie, I have a couple more.
Yes.
So on the ilium, I show the pubis, but the pubic crest and pubic symphysis. Oh, that wouldn't have been on. That would have been on today's picture, right? Of the ilium or the coxal pelvic bone.
Just put it on a list and send it to me. Let me take a look. Okay.
Okay. Okay. I'll try and put them all together.
I pointed it out today, but I don't know that it was labeled.
Okay. Okay. I may have missed
So
it.
what I would do is just put the PowerPoint, and up and add it to it.
Okay. Thank you.
Thank you, Charlie.
Bye bye.
Right.
Is it May or May?
Yeah, May.
May.
Thank you.
Thank you. I'll let you go.
Okay,
Have a good day.
bye-bye.
Bye, Charlie.